So our next presenter is Mert Basamachi. Forgive my presentation. And he's our colleague from Turkey. And he'll be presenting his work about using Bound-BERT-based model for protein-protein interaction extraction from literature. Go ahead.
Thank you. Let me share my screen first. Thanks for the introduction, and I also would like to thanks for the VDOS organization for giving us the opportunity to make this presentation. And welcome everyone. Here is our outline. In the introduction chapter, we describe some basic concepts that have been used throughout our study, including the problem definition as well. And methods and results chapter describes our overall study under the three main headings. First, we apply protein-protein interaction extraction, and then we extend our study for the host and pathogen interactions on the COVID-19 dataset. For that purpose, we created our own COVID-19 dataset. And later on, we extend our study for the possible drug discovery, which can be used as a potential application and use case for our research. And on the conclusion and the future work chapters, we will describe and we will make our final remarks. Protein-protein interactions play a central role in many biological processes such as cell metabolism, signal pathways, DNA replication and transcription, and conversion from genotypes to phenotypes. Considering their central roles in those processes, PPIs have been widely examined in many biomedical studies. Fortunately, these studies are mostly published and stored in literature databases such as PubMed and Medline. And also there exist some protein interaction databases and interaction networks that stores the current knowledge in the structural format. With the rapid increase in the biomedical literature, manual detection and extraction of PPIs from the long biomedical papers is a time-consuming and exhausting task. Moreover, keeping PPI databases and interaction networks updated also requires a manual effort. Therefore, there's a strong need for the automatic relation extraction and NLP techniques for that domain. Furthermore, the current coronavirus pandemic reveals the importance of such automatic techniques, since the ease of access to current biomedical knowledge regarding interactions of human and SARS-CoV-2 proteins can help researchers to discover possible treatments and drugs. Host and pathogen interactions are a type of interspecies protein-protein interactions, and they refers interactions between host cell proteins and the pathogen cell proteins. Moreover, interactions between SARS-CoV-2 and human cells are also falling to the HBI category. And another concept that we have used in our study is drug and target interactions, and they described as binding of a drug to a target location that results in a change in its behavior or function. Those drugs could be chemical components, medicines, while the biological targets are proteins, genes, receptors, or the enzymes. So let's move on to our problem definition. Actually, our goal is to determine whether pairs of proteins that appear in a biomedical sentence interact with each other or not. So let's move on from an example sentence. Here we have a sentence with three different protein entities. And if you look at the semantic meaning of the sentence, we can easily understand that LEC interacts with two proteins, CCR1 and CCR8 as well. And there is also non-interacting protein pairs, which are CCR1 and CCR8. And here our goal is to determine interacting and non-interacting protein pairs. This problem can be solved by several relation extraction techniques and NLP techniques, and previously several kernel-based techniques have been proposed. And recently, with the advancements in the deep learning area, there are several deep learning approaches have also been applied for the same problem. And how we solve this problem? For the protein-protein interaction part, first we have used 5 benchmarking PPI corpora, which have been proposed by the PySolo in a standard unified dataset format. We have 5 PPI corpora, which are AIMET, BioInfer, HBRD50, IAPAL, IAPA, and LLL. And this corpus each corpus has different statistics, as you can see on the right table. For example, AIMET contains around 2,000 sentences with 1,000 positive entity pairs and around 5,000 negative entity pairs, in total 6,000 sentences. And we apply some pre-processing steps for each sentences in our datasets. A sentence with number of 2 or more entities contains and chooses 2 different entity pairs. For example, in this sentence, we have 4 different entity pairs. Therefore, we can generate 6 different entity pairs. And for each entity pair, we replace exact protein names with the protein 1 and protein 2 keywords, and the other proteins with the protein keywords. And lastly, we have labels for each entity pairs. And we applied a transformer-based model, BioBERT, in order to solve the protein-protein interaction extraction problem. BioBERT is a pre-trained language model which is trained on a large-scale biomedical corpora, including 4.5 billion words from several PubMed abstracts. And it achieves state-of-the-art performance on many bio-NLP studies. Moreover, it can be fine-tuned by using custom data sets to perform different bio-NLP tasks. Here, we applied our own PPI dataset for fine-tuning to perform protein-protein interaction extraction. During the fine-tuning stage, we feed each sentences of our dataset to the BioBERT model, and the whole pre-trained model and its initial weights are updated during the fine-tuning stage, and we obtain a final model that can perform protein-protein interaction extraction. And for those who are interested with the details, these are our hyperparameter settings for the BioBERT model, and here are our results on the 5 benchmarking PPI corpora. We compared our model with the previous studies, and BioBERT model achieves a state-of-the-art performance on one of the biggest corpus, AIMET, which is 79% F1 score. We obtained 2.1 F1 score improvement compared to second best study. And we have also obtained a comparable results with the previous studies on the other 4 corpus. And for example, on the BioInfer, we obtained the second best, and for the HBRD 50 and IAPA, we obtained the third best scores. And later on, we extend our study on the PPIs for the host and pathogen interactions. First, we created our own dataset for that chapter. To the best of our knowledge, we proposed the first golden dataset for SARS-CoV-2 pathogens and human host proteins. Dataset creation steps can be summarized as follows. First, sentences are extracted automatically by using an in-house biomedical literature mining tool, SciMiner. SciMiner uses gen and protein entities obtained from Hugo and for the host entities, and it was also extended to identify coronavirus proteins and genes by utilizing the ontology of genes and genomes, OGG, for the coronavirus entities. And then after we automatically extract sentences by using the SciMiner, We also performed some manual annotation steps. In this phase, 3 biomedical experts are involved for the annotations, and annotators manually filtered out some incorrectly labeled entities first. And then each of them individually labeled whether host and pathogen pairs interact with each other or not. Later on, they come together and evaluate each sentence and interaction labels together to solve conflicts and disagreements and achieve a consensus on the annotations. Finally, created COVID-19 datasets contain 94 sentences with 64 positive interactions. And then we applied our BioBERT model for the HBI extraction task. First, we combined 5 benchmarking PPI datasets into a single data source and fine-tune it on this data source. And then we applied our model for the HBI COVID. We applied it for the HBI COVID-19 data set, and we obtained a very promising results on the HBI extraction, considering it is a interspecies interaction extraction. We obtained 65.8 F1 score with a very high precision, which means we have 0 false positives. And lastly, we extend our study for the possible drug discovery by combining our HPI network and drug target networks. Actually, it is a possible application for the drug discovery, and actually it is a use case. It can be used as a use case. First, we created a host and pathogen interaction network by using our annotated HPI entities, and then we combined this HPI network with another study, with the outcomes of another study on the drug and target interactions. And we found that the drugs targeting the host pathogens can possibly be used for the treatment of the SARS-CoV-2 disease. Here you can see our combined HPI and DTI network. Oranges are our host proteins and greens are our pathogens. And if we merge our host and pathogen interaction network with the DTI network, we can reveal possible drugs that targets our host entities. And many of my results are represented in the coronavirus infection disease ontology. And lastly, we would like to mention ontology applications in our project. Coronavirus proteins and genes were identified by utilizing ontology of genes and genomes for coronavirus, OGG. And literature mined results can be modeled, manually verified, and then represented in coronavirus infection disease ontology. And as a feature word, we will also include the interaction types by using interaction network ontology. And in the conclusion part, we would like to mention our contributions in this research. We have 4 main contributions. First, we showed that a BioBERT model can achieve a state-of-the-art F1 performance on AIMET corpus, which is one of the largest corpus, and it also achieves the comparable results with the previous state-of-the approaches on the other 4 corpora. Secondly, to the best of our knowledge, we proposed the first manually created dataset for host and pathogen interactions on COVID-19 dataset. And third, we extend our study by applying fine-tuning the BioBERT model on our golden COVID-19 dataset for host and pathogen interaction extraction. And lastly, we merge our drug target interaction network with the created host and pathogen interaction network to reveal possible drugs and treatments for the SARS-CoV-2 disease. As a feature work, we would like to incorporate with the other ontologies rather than just using only OGG, such as we would like to use INO for the interaction types, relation types, and we will also like to include the coronavirus infectious disease ontology. Moreover, we would like to expand our HBI dataset and train our model on that HBI dataset rather on training on the PPI. And lastly, we would like to extend HBI study on the other pathogens rather than not only using coronavirus pathogens. Here is our contact list, and if you have further questions later on for this session, you can always email us. And I will also give a special thanks to my professors, Arzucan Özgür, Yunkun Kur, Yunkun Oliver He, for letting me involved for their valuable research as a master's student. Thank you very much. Thank you very much. If you have further questions about this study, we could answer on the remaining part of this session.
Thank you. Any questions? I have one question. So, protein-protein interaction for NLP is not new, right? So I'm just wondering, for like protein-protein interaction on COVID-19, is there only like a special situations you have to handle, like special challenges you're facing during the implementation?
Mm-hmm. Yes, actually, a protein-protein interaction dataset that we have used for training our model is different than the host and pathogen interactions. Actually, these are two different domains, but we would like to investigate how a BioBERT model trained on the PPI corpora is successful on interspecies interactions. And luckily, we obtained a good results on the HBI dataset. However, we would like to mention that we trained our model on the PPI and then we tested on the HBI. Actually, one of the most challenging is that our own created HBI dataset is very small. Therefore, we couldn't train our model on that HBI dataset. Actually, we collected the HBI information until to some PubMed articles until to June of 2020, but in the future we would like to extend this study for collecting more host and pathogen interactions, and we would like to investigate how BioBERT model achieves and how BioBERT model is successful on this HBI specific data. We believe its performance will be increased when we trained on this special HBI data.
I see. Thank you.
Thank you.
Any other questions for Mert? Oh, Yuki, you have a question?
Yes, I have one question. What terms were included in the false positive? Do you have any tendency or not?
Do you mean for the host and pathogen interaction extraction part, or for the protein-protein extraction part?
Extraction. Both.
Actually, having a high precision system for the relation extraction studies in this domain is very important. And actually, we didn't mention false positive rates in this table, but to the best of our knowledge, BioBERT model achieves a good precision on relation extraction task, which is very important for the biomedical domain, I think, because if it states that if there is a interaction between two entities, most with a high ratio, it is correct, and I could say we got a high precision system with low false positive ratio.
Thank you.
And one of the interesting parts here is that on the HBI dataset, we got 0 false positives. We found very interesting, which means our model works, and Biobert model works very good on also other data sets.
Thank you. And what is a false negative included? What is that?
I'm not sure I could understand correctly.
What
Well, I
terms
think, yeah, she's asking about why the recall, like if the recall is low, then why, I guess, right? You think that's the false negative, right?
that I wanted to say.
Yes, because of it, yeah, we got the best recall scores on the one of the largest corpus on AIMED. Since the false positive rate is small, I think our recall rate is also
So basically,
one
if
of
you
the
want
best.
if you want to achieve a high precision, you have to sacrifice your recall a little, right, to balance it. Yeah. Okay.
Okay.
Yeah. Thank you again. I think this is a great work and very important for the next step, I guess. So let's move on to the next talk.